From Microbe TV, this is Q&A with A&V. I'm Vincent Racaniello, and joining me tonight from Chevy Chase, Maryland, Amy Rosenfeld. Hello, Amy.
Hello, Vincent. How are you today?
I'm well. How are you?
I'm good, except there's flood warning watches.
And happy birthday, Amy.
Thank you.
Amy's birthday was yesterday, the 9th of August. Everybody wish Dr. Amy a happy birthday, please.
How are you playing in the background?
What? What's wrong, Amy?
I hear the repeat in the back of my AirPods. I don't understand why I hear the repeat.
I don't hear anything. I'm sorry. Anyway, yes, we're Team Purple tonight, right?
Yes, Team Purple.
I got my usual button-downs, and Amy has her, I don't know, is it cashmere? That would be my first guess.
It's always cashmere blessed. It's always cashmere.
Anyway, I'm not going to say how old Amy is. It's not right to say anyone's age. But happy birthday, Amy. I hope you have many more.
Thank you very much.
It's been a pleasure.
Look at
all the happy birthday wishes. Oh my gosh, you've got like hundreds.
Yeah, it's great. Thank you so much.
They like you. It's only a day. It's a day later. Anyway, welcome to our Wednesday, our midweek virology little corner of the internet. I don't want to call it office hours because not everybody knows what that is. It's a place to talk about viruses with us and amongst yourselves. And our wonderful moderators are here tonight. Let's thank them. We have Les, we have Steph, we have Frank, we have Tom, and we have Vanity Nutrition. And if you think that viruses are ending with COVID, or I should say virus stories, you have seen in the past few weeks that it's not the case. So, Amy, are you hearing echo still?
Yeah, it's total repeating in my AirPods, like total repeat. It's a loop.
I'm sorry.
How do I fix it?
So the AirPods are set to be connected to your Mac, right?
Yes.
And you went into the system preferences and you made sure that they're there, right? Go back in and make sure that the output is the earbuds. Okay. Let's see how that works.
Yeah, it's the AirPods. Yeah.
What's the input?
My AirPods.
There shouldn't be. It should be your microphone there. Your input should be the Samsung Q2U.
Oh, okay. Now it's better.
Okay.
No, it's not better. Not better.
Tap the mic, please. We should have done this before we started. See, the mic is not on. You sure the mic is selected in the system? Is it turned on?
Hold a second. Yeah, hold a second.
Okay. In the meantime, while Amy's sorting that out, I just wanted to show folks this cool T-shirt. Where is it? Yeah, there it is. So that was given to me by Dr. Jackie Mayo, who is a internist in New York City who visited the incubator last week, and that was the title of a very recent TWIV. Hello, Amy. How's the mic? Yeah, the mic is working now.
Yeah, but I still hear the repeat in my headphones.
Well, do you have the original headphones I gave you?
The red ones?
No, the headphones that
No, those are those I left in the office.
Oh.
Never took them with me.
All right, so it's impossibly repeating for you, or you can deal with it?
Yeah, it's kind of annoying.
It's got to be something to do with the way you're routing the audio, you know. It's probably
I don't know.
There's a little bit of a delay on the earbuds, so I don't know what else to say because it's all set in the preferences, right?
I don't know. We'll figure it out later. It's just kind of crazy, but it'll be fine.
All right, so let's start with our... Someone wished you happy 21st birthday. Makes you feel good, right?
It's good.
Amy's light, your light is looking good. Thank you, Richard, for the lights. It's looking good there in the background. Maybe you could take it down a notch with your fancy remote there. Is the remote on? There you go.
Yeah, I turn it on.
There you go. That's good. Perfect. No, not too much. No. Put it back up. There you go. Thank you very much. Okay, let's do some questions. Are you ready for some questions, Amy? A lot happening in the virus news this week. But first, to what extent can a virus affect behavior? Could there be some
what behavior?
Any host behavior, let's say. And in this case,
like
Billy
rabies
Bob.
behavior?
Pardon me?
Like rabies behavior?
Yeah, so rabies makes people behave differently, right? But in terms of adventure, careless, or excited, afraid, I don't know that we know that. Let me give you two examples of viruses of other hosts, though, that change behavior. There is a virus that infects ants, and it makes the ants grow these things out of their heads, and so the birds see them, and they pick them up, and they spread the virus elsewhere. There's also a virus of caterpillars that makes the caterpillars crawl to the top of the plant, which they normally would not do, where they could get eaten. And then they explode and they spread virus all over the leaves, so it's easier. So those are behavior-changing viruses. So whether that happens in people, hard to do the experiments, right? Now, MK says we'll be discussing the longivirus from shrews. So, have you heard of longivirus, Amy?
Yeah, I think I sent you the paper.
So let's see. I have a little article here from ProMed Mail. Here's a New England Journal article. A zoonotic henipavirus in febrile patients in China. So basically, a henipavirus is a genus in the paramyxovirus. Hendra and Nipah are henipaviruses. So this is related to them. And they say during sentinel surveillance of febrile patients with a recent history of animal exposure, they found a virus in them. And 35 patients with acute infection, no other pathogens present. They had fever, fatigue, cough, anorexia, myalgia, et cetera, thrombocytopenia, all sorts of things. And seropositivity in goats and dogs. And predominantly in shrews in the wild. So they may be the natural reservoir. So it's associated with the people. It's associated with the people. It's not clear what it's causing, but we'll find out, I'm sure. Right? Okay, so what do you think, Amy?
What about it?
Yeah, we don't know anything about it yet, but it's there, and it just goes to show that if you look, you will find more viruses, right?
Yeah, for sure.
What are your thoughts on the cardiovascular preprint of Thailand showing elevated troponin after the second Pfizer dose in 13 to 18-year-olds in 5 out of 300 participants? So you know what troponin is?
I don't really have.
It's a muscle.
Yeah, it's a protein.
Right?
It's a muscle. It's a muscle protein, and like how the the Z bands move and stuff. Not clear what it means.
It may be that
I didn't really read it.
It could be. Well, we know that myocarditis is associated at a rate of 1 to 20 out of 100,000 young people. So it may be that there's some heart muscle damage and it releases troponin, right?
Yeah.
I actually found an article on it. This is actually found before. They called it troponemia because it's in the blood. So I think it's possibly associated with the myocarditis. And maybe there's heart damage in more people and not resulting in myocarditis. Could be something like that.
Maybe.
Any thoughts on trying to dodge COVID? Doctors don't want to give anyone Evusheld. It's unfortunate because Dr. Mayo said to me last week, it's very easy to go to get. She gives it, and she said,
in New York it is. Maybe
I
it's
suppose, yeah.
not.
Maybe elsewhere it's not. It's true. But how to dodge? If you want to dodge, you take Evusheld because that's a preventative, right?
Yeah, I guess. Man, I'm not sure that I agree with that, but okay.
You don't agree that it's a good preventative?
I think that everybody's going to get infected, and so you should just accept it and move on.
Some people don't want to be infected, right?
Well, some people don't want to get old.
That's a futile fight, isn't it?
Exactly.
Oh my gosh. I know someone who wanted to extend his life 10 years.
Yeah, I'll give you $1 million if you could tell him how to do it.
I don't know how to extend your life for 10 years. You know, I just don't. If I knew, I would give it away. So then we're in trouble because everyone lives a long time and the world is already overpopulated as it is.
Well, we're not made to live a long time.
Here's a follow-up. Well, we already live longer than we did before, right? We used to have an average life in our 30s, and now it's in our 70s and 80s. And
yeah, it's called medical intervention.
And then we develop Alzheimer's and cancers that we didn't before, right? Because we were young dying. So what do you want? You want to live older and get horrible diseases or what? It's a tough choice. Is it as deadly, the new Langia, as Nipah and Hendra? It doesn't seem to be. These are patients who are not dying, right? So there's no evidence that it is. But I think we need to see what more information comes out of there so far. Okay. Is there any truth to the theory that toxoplasmosis in humans can cause fearlessness? Not that I'm aware of. And it makes mice, toxo in mice makes them lose their aversion to cat urine. So they're no longer staying away from cats so the cats can eat them and get infected and spread it. But you should go listen to TWIP, This Week in Parasitism. Do you think Dixon would like to do a parasitology live stream, Amy?
For sure.
He and Daniel, it would be fun, wouldn't it? The two of them on a live stream. It would be a hoot.
Yeah, it'd be good.
Maybe we should live stream TWIP.
Okay.
That doesn't sound enthusiastic. What do you think?
Well, the thing is Daniel has said that he would do a live. He's like into the live stream, but yet he's only done one live stream for the COVID update or the TWIV clinical update. So.
Well, I have to push it, basically. When I push it, he'll do it, Amy. So, because I'm the... Did you know I'm the producer?
You're the producer?
There was a movie with Zero
and how's
Mostel,
that going
right?
for you?
Or there was a play or a musical with Zero Mostel?
The Producers was a movie and then became a Broadway play, yes. Everybody knows Mel Brooks' movie,
that's
The
right.
Producers.
I didn't see it.
Hitler on Ice.
I did not see it.
You never saw The Producers?
No, and you never saw Star Wars. Okay, so we're even. What do you think? Hello?
Yeah, what? I'm trying to process how you made them equivalent. I am going to need heart palpitations. I might need
so here's Midlands man from Ireland. Would you anticipate more widespread detection of polio in sewers around the EU-US if it were been tested for? Or are current cases rare? What do you think?
Would you anticipate more widespread detection of polio in the sewers if it had been tested for? What is that? I don't understand the question. I've always thought that polio was in the United States.
Where are you going?
Changing the air conditioner. It's too cold. So, I mean, the truth of the matter is, I don't understand the question, but
Well,
there
can I
would have
rephrase?
been,
Can I rephrase?
sure,
If we look more, are we going to find more widespread polio in sewage?
for sure.
Yeah, I agree. I think it's been there all along, as we've said for years. The cases of paralytic disease are rare, fortunately, right? Because a lot of people are vaccinated and it only paralyzes 1 in 100 or 200 infected people. But it's always been there. And the more we look, the more we're going to find. Every
Right.
city you look in, you're going to find it. And people are going to go freaking out when Amy and I have said for 5 years, seek and ye shall find it.
5,
Longer?
way longer. 2015, we talked to Mark. Yeah.
You guys should
So
look
get
up
this
that
one.
2015. Oh, it wasn't on a TWIV. It was just we talking to him, right?
Yeah, at ASV. He sat
Mark
behind
Palance
us.
used to be the head of coronaviruses at CDC. Is that right? If that's if
Polio
that's
lab.
if that's a thing, yeah. And he said we don't look because we're sure we'll find it, and we don't know what to say when we do.
Right. So I was on this podcast on
You were,
Friday,
yeah.
and they had this pediatrician, and they asked her about the polio vaccine, and she said, "Oh yeah, I tell my patients up until the outbreak that it was one of those vaccines that it was not the most important vaccine at all." And I was like, "Are you kidding me? It's the most important vaccine because that's how you control polio out from when we no longer see out polio. Like
Yep.
seriously?" Not
Yep.
a good statement by the pediatrician.
Do you think the new RSV vaccine Daniel was talking about will avoid antibody-enhanced disease the old one is suspected of? Do you know what this new vaccine is based on, Amy, offhand?
It's the post fusion it's the post
Prefusion.
fusion
Prefusion.
pre oh, I thought it was post fusion, but yeah, maybe it's the pre-fusion G protein. It's just like the one they did for um COVID SARS-CoV-2. I think, and it's mRNA based, I think,
Yeah,
or
I
at
mean,
least one of them.
one of them is. The problem with the original vaccine, it was an activated virus. And one of the problems was the inactivation procedure destroyed the F protein, so you didn't make antibodies against it. And so when you got infected, the virus was able to fuse and move from cell to cell via the F protein. That's one of the mechanisms. It also messed with innate sensing. There were a lot of issues with that original vaccine.
Well, that's the problem when you make an inactivated thing. It's you just treat it with formaldehyde, and you just hope that that the epitopes remain intact. It's not always
Yeah.
clear that they do.
One thing, virus after another in the news. Yeah. I mean, I think we're sensitized to viruses now because of the pandemic, because these little things didn't make as much of an impact before, but now one after another. So we have COVID, we have monkeypox, we have polio, we have this new virus in China. Every little report makes big headlines, right? And you know the reason, Amy? Because everyone wants to pick up the next pandemic.
Well, it's not just that. It's the fact that having been a pandemic and went home, like we've screwed around with all of our natural immunity and resistance to what's ever going around in the environment.
Yeah. So here we go. Does poliovirus infect vaccinated folks?
Yes.
If so, and yes, are there any symptoms in vaccinated people
There are no
infected with
symptoms
polio?
in most unvaccinated patients.
Yeah.
So why would it start to make symptoms in vaccinated patients?
Right. So no, there are no symptoms. Even though this is a gut virus reproducing in the gut, you have no gut symptoms whatsoever.
No, and when somebody told me to try and spin it to get a job as a gut virus, I looked at them and I was like, seriously? It doesn't cause diarrhea.
No. But the vaccine does not prevent infection. Neither the IPV or the OPV does not prevent infection.
No vaccine prevents infection. Why would this be different?
It should be a T-shirt. Vaccines do not prevent infection. And people will look at it and go, why are you wearing a T-shirt that says that? And that's when you should listen to the live stream, because that's really an important thing, right?
Super.
Here's one I don't know the answer to. How do people develop Wagner's autoimmune disease? Do you know what that is, Amy?
No.
Let's look it up. It's a hereditary eye disorder that leads to progressive eye vision loss. That doesn't sound good. I don't know what's the etiology of it at all, but I can almost guess that it's not a virus. What do you think?
Not a virus.
How effective is the flu shot this year? Well,
I don't know. We're not at flu season yet.
We're not, but although down in the southern hemisphere they are, do you think there might be some reports from down there yet? Effectiveness of flu shot, southern hemisphere. Let's look. Yeah, nothing coming up quickly. I don't have any information from down there. Happy birthdays streaming by here. Treat yourself. Here we go. Treat yourself to a new pair of Manolos.
Okay.
Would you say we are 10% human and 90% bacteria?
Sure.
This number was revised, right? Do you remember that? We talked about that on TWIM, actually. Bacteria make up 1 to 3% of body's mass. Okay, so that's it. It's not 10% human and 90% bacteria. It's more like...
Maybe it's the genetics of it that they're thinking about, not actual mass.
Yeah, in terms of mass, it's not. All right. Oh, Tom's wife has the same birthday as you. Tom is one of our moderators, Amy.
Cool.
So the 9th of August.
Cool.
Why doesn't Amy like plants in her house?
I don't do dirt. No dirt.
So Amy doesn't do dirt. But you don't even want plants in the incubator, right?
No plants in the incubator. And definitely no fake trees in the incubator that then you have to dust. That's the most ridiculous thing in the world. Dust your tree.
So we have a fake tree here.
What do you do? Who do you hire to do that? The Dust the Tree Company?
Dust tree busters.
I see.
We have a plastic tree in the house here. And my wife said the other day, get rid of it. And I said to Amy, can I bring it to the incubator? And she said, no. But I said, but there's no dirt. She said, no fake plants either. But the leaves get covered with dust because you have to dust them. It's kind of ridiculous. There's a real plant I don't think gets dusty, but this one gets dusty. It's crazy. And I do not wish to dust a fake plant. I always found engineering quite interesting and an important subject as it is, but until I've now begun to realize how much biology is such a profound and almost unfathomable study. Biology is very cool. I don't think it's unfathomable. I think parts of it we understand. Don't you think, Amy?
Sure.
Yeah, I think a lot of it we understand. And to follow up on that, Amy, what got you into virology? I'm going to say Vincent Racaniello, but you can answer it yourself.
That's close. Yeah, that's cool. Yeah, that's pretty much part of it. Yeah.
Yeah, Amy came to do a rotation in my lab. What year was that? 1991?
99.
99. And she ended up doing virology. And there you go. And then she turned out to be my last trainee. Amy, do you know anything about psoriasis?
It's an autoimmune disease.
It's an autoimmune disease. That's right. But if you want to know more, you need to listen to Immune. I don't know if we've done an episode on that, actually. Here we go. Our pharmacos from, that's not the right word, pharmacists from San Francisco.
Yeah, Kip and Laura.
Kip and Laura. Hello, Kip and Laura. That's Kip.
It's Kip.
Monkeypox asymptomatic spread. I think that's one of the areas that that's unknown, right? We don't know if there can be asymptomatic spread. So I think it's a good question. If you go to the CDC website, they admit that we don't know. Would you agree with that, Amy?
Yeah, I think so.
So yes, if you're a dentist, that would be of concern, so that we need to find out ASAP, right?
Yep.
Now, here's some interesting news from the UK. UK children between 1 and 9 are to be offered a polio booster if they live in London. What do you think about that, Amy?
Why are they offering a polio booster again?
Why are they offering? Oh, just because, in a certain neighborhood, a good fraction of kids were unimmunized. So it seems that they just want to cover them all in case they haven't been immunized, right? But
it's no different than what they're doing in Rockland County, except the fact that most likely they're both giving the wrong vaccine.
All right. You want to give OPV?
Yeah.
And I want to give IPV.
Yeah, well, that's not a good plan.
Why?
Because your guts are fully susceptible, and without exposure to infectious virus, you don't get a good response to begin with.
Yeah, I mean, I understand that, but I think we also don't want to reintroduce Sabin.
It's already here. What's the difference?
I knew you would say that. Yeah, it's a good point. It's a good point. Anyway, this is good to be offered. Actually, they should find out their vaccine history and give them a full course of 3 doses if necessary, right? And if you're an adult and I've gotten a lot of emails this week from adults who said, I was never vaccinated. Should I get vaccinated? And I think the answer is yes, Amy, right?
What's the question again?
If you're an adult and you were never vaccinated against polio, should you get vaccinated?
Oh, for sure. Yeah, but why weren't you vaccinated? Because you took somebody's advice who told you that it was one of the least important vaccines?
I don't know the reasons. They often don't say the reasons. Oh, some of them do say that I was part of a religious community that had no health care, and now I'm not part of it anymore, and I want to get vaccinated. So I say, yeah, get vaccinated. Is hepatitis and malaria worsened by the climate crisis? All right, so
I would imagine malaria. I don't know that hepatitis is that way. What's the vector?
Well, there are multiple viruses that cause hepatitis, right? None of them are
but A, B,
driven.
and C are the main ones, right?
Yeah, there's a lot of delta, and there's also E, Hep E. There's quite a bit of Hep E, but none of them are vectored. So that first of all, malaria, of course, is mosquito vectored. So as temperatures rise, the mosquito range increases, and so the malaria belt is going to widen. That's very clear. But even for a non-vectored virus, Amy, one that goes from person to person, is there some way you can imagine that climate increased temperatures could impact the disease in some way?
Sure. I mean, polio goes person to person, and climate affects its spread.
Yes, so warm climates benefit polio spread. So it could very well be that these viruses will react in the same ways.
So did you know that I'm unaware that there is a polio pandemic right now caused by the OPV vaccine someone got in another country, which means giving more OPV is a bad idea and it might be a resistant strain. Did you know that? Did you know that I was ignorant?
The comment was probably removed, Amy. You shouldn't say if it's an offensive comment, there's a good chance that it's going to be removed. So wait till we get to it. I don't know
anybody would find it as offensive as I do.
No, I agree. And then second to you would be me. right? Because I trained you and I know what you know.
Yeah, exactly.
Is it possible for to make a vaccine for cavities?
Yeah, it's a bacteria, sure. But I don't think it's happening.
Yeah, this is out of my lane, and I don't like to go out of my lane. So I would, yeah, it is bacterial, but it's part of your normal bacteria in your mouth, right?
Sometimes not.
Okay. Okay. What else here? On the fence to get a second booster or wait until BA.4/5. By the way, Emma Thompson, the virologist in Glasgow who has the preprint about AAV and the pediatric hepatitis kids, she emailed me, and because she's going to be on TWIV when I go to Manchester in September. She said, by the way, we call it BA.4/5 because the spike is the same in both. And so that's why it's for both. Whatever vaccine is made is going to cover both of them.
Okay.
Okay, so are there any data showing the enhanced vaccine is clinically superior? No. The only data we have are that the Omicron specific vaccine, and that's 1, 2, not 4, 5, gives you an increase in antibodies against those viruses. But that's all we have. We don't have any clinical data, anything about disease. So that's what you need to make your decision on. Nothing else is going to be coming available. And if you want a booster, then I would wait till 4 or 5 comes out. I'm not getting one. And I don't think Amy is either.
No, probably not.
Well, this is an interesting one. If I had yellow fever 3 years ago, is vaccination required for me not to get it again?
Probably not. It's probably a boost.
I don't know how durable immunity is after natural infection. That's a really good question.
Has to be as durable as after vaccine, or
You think
even
so?
better, for sure. I've yet to find a vaccine that gives better durable immunity than natural infection.
I don't know. I'd get the vaccine anyway if you're going to go to a yellow fever endemic country, just to be sure.
Well, you're just stimulating that memory response. So
Yeah.
it's fine.
Oh, so Amy, you sent me a preprint, or maybe it was a paper, saying that in older people it takes 6 months to get a full development of the antibody response.
I don't know that they said 6 months, but yeah, that was me.
So,
but then they said it was more sustainable than anybody else's. So,
but the problem is, if you get infected before that time, then you die.
You don't die.
Well, the majority of people dying of COVID are
but a majority of infected people
over 75. So,
but that's an irrelevant. That's an irrelevant statistic. If I take 1,000 80-year-olds, 4 die. That's the statistic, not your statistic.
No, I'm just trying to understand if that explains why, even if you're vaccinated, if you're over 75, you're more likely to die. You don't think it explains
more likely to die
of COVID? I mean,
I don't think you should say that. I don't think that.
Well, that's what Daniel said. He said the vast majority of people.
But that is the number of cases that he particularly says as an anecdotal thing. If you say, I'm going to survey the entire community of Great Neck, Long Island for 70-year-olds, I bet you will find 75% of them are infected or have been infected.
Okay.
And 2% go to the hospital, and then 0.01% of that 2% die.
Okay. Are upper respiratory infections usually viral, while lower are bacterial? Well, infections versus disease. I think probably the question is about disease, don't you think?
Probably.
Because, both the upper and lower tract are colonized by bacteria. It may be correct but there certainly are viral infections of the lower tract. When you think about the
Yeah, I
serious
don't know that I would say that.
when you think
I
about the
don't know that
serious
you could say
pneumonias,
that.
you have lower tract bacterial infections, right? With Amy, it's always difficult to make such generalizations.
Yeah, I'm very picky.
All right, one more from Touj. You're asking, it's good to ask questions. Do you support the idea of having kids play in the dirt to benefit their immunity? What do you think, Amy?
I do believe that you should not. Is that like asking if you should like spritz your kid with Purell?
No. Let kids play outside so they pick up a good
Yeah, I think it's
microbiome.
good to let kids play outside.
Yeah, I think it's fine. Yeah, they're good. They're good. Some good evidence that kids who are exposed to the environment have developed strong immune systems.
Hmm.
Are you still getting echoing in your earbuds?
Yeah, it just repeats. The whole conversation's repeating. It's not echoing. It's a repeat of the whole conversation.
Oh, you have to turn off YouTube. Do you have YouTube? Do you have the stream playing in another window?
I don't think so. Hold a second.
Just close your browser. Close Safari.
Okay, I closed Safari.
And is it still echoing? Hello, Amy? Amy?
No, I don't think so. I think it might have gone away.
All right, good. Okay. We got a lot of suggestions here. Okay, good. Can you comment on adult sudden death syndrome attributed to the recent death of 5 physicians in Ontario with a possible link to COVID vaccination? No.
I don't know anything about this. I never heard of this.
I doubt it's a link to COVID vaccination. It's something else that we're not aware of. But we'll see if and when the paper is published.
Oh, there's never going to be a paper.
Probably not. Can you talk about this new intradermal vaccination strategy for Genomes? Amy sent me the press release today. Go ahead. Tell them about it. Would you like to?
No, you should.
So Genomes is normally given subcutaneously. They did a trial to see if intradermal would give the same immune response because you use 1/5 of the amount of vaccine when you do intradermal. And according to the FDA, that you get the same immune response in terms of neutralizing antibodies. So they gave an EUA, an emergency use authorization for Genomes for intradermal vaccination. So that means we're going to have more vaccine available because the supply of Genomes is limited.
But you're having the guy on Twiv on Friday.
Yes, so Michael Merchlinsky, who is at BARDA, Biomedical Advanced Research Development Agency. Is that what it stands for? Let's see. BARDA. Biomedical Advanced Research and Development Authority. Okay. He's going to explain all about the development of these smallpox vaccines and the smallpox antivirals, and we'll certainly ask him about this. And before I take another question, I just wanted to mention to folks that we do have a TikTok channel now. We have two of our interns creating little TikTok videos. So let me move it so you can see the link here. So here is.
Scroll down.
Yeah, you're right. I have to scroll down so we can see the link, right? Unfortunately, it's tiktok.com/microbetv. Can you see that? No.
So you should scroll down.
No, it's a weird business with the sharing. Here you go. Thank you. There it is, tiktok.com/@microbe.tv. Okay? And then you see all the short videos, including some of me and Amy. There's just 2 up so far of me and Amy. There's one new definition for pandemic, and Amy pimps Vincent. Okay? So,
Yeah.
TikTok. We're doing this to try and get more engagement of a different type of crowd. And so far, these are being nicely liked. I'm putting some of them on Instagram as well. Okay, so, and people over there are like, wow, I have to get TikTok now.
It's good.
How does the rare but regular integration of Hep B DNA into hepatocyte genomes have any significance to chronic infection? I think it's irrelevant because it's not part of the reproduction cycle, right? It is the viral DNA is episomal, closed circular DNA, which reproduces separate from the chromosomal DNA. So the integration is not a consequence, although some people think it has something to do with oncogenesis, liver cancer. But that's a bit of a controversy. Any thoughts on the theory that adenovirus is causing liver failure in these pediatric cases? So there are
Emma Thompson.
Two preprints are now out saying it's adeno-associated virus. Emma Thompson is on one of the papers, and there's a second paper. We're gonna have Emma on TWIV in September. I'm going over to Manchester to a clinical virology meeting to do that, and she'll talk about it. But you need adeno-associated virus needs a helper virus to help it to reproduce, and it can be an adenovirus or herpes virus. And in fact, they find adenovirus and herpes virus in all these kids as well as AAV. So it looks really interesting. Do you think monkeypox could be a big problem for kids once school starts? A lot of people are wondering about this, right?
Mm-hmm.
I think any kind of sports where you have extensive contact, like wrestling, can be a huge problem. What do you think, Amy?
Could be. I mean, it's not as easily spread as smallpox, so could be, but I don't know.
So wrestlers, have close, sustained skin-to-skin contact, and so if you have a lesion.
Well, hopefully you're smart enough not to be wrestling if you have a lesion.
Yeah, I don't know what the solution is. So as many people tell me, I don't know anything about public health, so I will not make any suggestions. But maybe there's a solution that involves somehow modifying behavior, because we're not going to have vaccines ready for school kids in the fall. But yeah, I think it can be a problem. Yeah. What will it take for a new variant to be named the Greek letter after Omicron, which is, of course, Amy, pi?
Oh, it's good.
It has to be substantially different in not just the spike, but in other genes as well. And the WHO, so the genetics is what defines whether it's another variant. And then WHO will say whether it's a variant of concern or not, based on what they think it's going to do in terms of resistance to monoclonals and so forth. Sorry, go ahead, Amy.
So I think that there is a new variant in India that they're watching, and it's not called pi.
What is it called?
I don't know, but it's not called pi.
It's a sub variant of Omicron called BA2.75.
No, it has a Greek letter name.
I'm not finding it.
I
Centaurus?
think it was on the... Yeah, that's
BA2.75.
Oh,
I don't
maybe.
know why they're calling it Centaurus. They make up their own names.
I don't know.
Rabies makes dogs bite. Yes, it does.
Well, some dogs bite irrelevantly of rabies.
I hate being at work when you guys are on. I'm sorry. It's probably distracting. If I had smallpox vaccination 40 years ago, am I able to get the monkeypox?
No.
Probably not. Now, I did some research today, Amy, on this issue.
Okay, I'm sitting down. I'm waiting for this, considering the fact last week
I know. We
You
had a
were
little.
wallowing in your wrongness. So
Okay, look,
I'm
I was thinking
waiting
about it.
for this one.
I think that so. In 2003, there was an outbreak of monkeypox in the U.S. caused by importation of rodents from, I think, Ghana. Okay. And I think there's a couple of papers on it. And I think around 38 cases, 7 of those were vaccinated with smallpox vaccine before 1972. Okay. So that's a long interval. And they said, so these people still got monkeypox, but it wasn't a serious disease, and so it could be that having the vaccine modifies the disease severity in some way, right? So either you don't get infected,
or vaccines do.
Yes, you don't get either you don't get any disease or you get less severe disease. That's absolutely right. Now let's talk about the other issue
But
from
that
wallowing.
says that you're still immune.
Yeah. So when I said
immunity still waned, so immunity was still present. Now, whether or not it's 100% protective against the development of severe disease, you have to define what severe disease is and what was severe disease back then. So I think it's fine. Yeah, I'm
so
fully
I think
confident
that
you
the
have
general
immune protection.
the general idea is that you are protected. Now, there is some. What did I see? They are recommending that certain people, even if you are vaccinated with smallpox vaccine, get vaccinated again. Let's see if I can find that. It's important to bring that up.
Why is probably another misinterpretation of crap.
Prevention. That's an Amy term, misinterpretation of crap. Vaccines, here we go. Vaccine protection. Just so people who aren't vaccinated, they're recommended. I saw some recommendations, on healthcare workers or other people who should be vaccinated. But I think if you are just an ordinary person like me and Amy, were you vaccinated, Amy? No, you were not. I was.
Oh.
So you're probably not at risk for contracting small monkeypox, right?
Right.
So I think. Now, can I just amend my wallowing in wrongness? Okay.
Wrongness, yeah.
Let me just chat a few minutes on this. So in my lectures, in my virology lectures, I have a slide that says requirements for eradicating a virus. Okay. There's 2 on the slide. One is humans are the only reservoir, right? And the second is infection or vaccination gives lifelong protection, all right? And that's where I got tripped up because I said, if you're over 40 years with smallpox vaccine, you're no longer protected. But I think Amy's point was it probably protects you against severe disease and death. So it's still protecting you even beyond 40 years. So that's it.
Exactly.
You have to think
Therefore,
of this
you have lifelong immunity.
Exactly right. Exactly right. Exactly right. I think they stopped at Omicron to minimize the panic. There will never be a pi variant. From now on, everything will be a subvariant of Omicron. I don't think so. I think that if it diversifies sufficiently, it will be another variant. But so far, it hasn't done that. Yeah. I don't know why.
Maybe if it diversifies too much, it doesn't use the receptor and it's dead. So
yeah,
not so good.
Non-infectious. 10 monoclonals tested against pseudovirus, bebtelovimab, efficacious against all Omicrons. Preserved essential antigen. Can they find this with vaccines? Well, it's a special site on the spike that is not changing in the variants, right? So yes, it would be nice to check that to see if you can induce antibodies, and that's one of the things that the Bjorkman strategy is doing. Yeah, these more conserved regions. Did fever mitigate SARS-CoV-2 replication, or maybe attachment to the receptor? I'm not aware of any evidence for that. Are you, Amy?
No. Plus, fever occurred after SARS-CoV-2 replication, so no.
Yeah. Do you think considering how similar many hantaviruses are, that drugs designed against Nipah or Hendra would be useful. I don't know. It's possible. Depends what the target is.
I don't know.
I mean, the drugs are not targeting the G protein. The G protein would be a target of a monoclonal or a vaccine, right?
Exactly.
But I have to say,
I think what's interesting.
Go ahead.
Would be if the horse vaccine protects against it, because it's a horse vaccine against Nipah, or Hanta, if I remember correctly.
Yes. So that's what I was just going to say. So there's a horse Hendra vaccine, which is the glycoprotein, and it protects against Nipah, and it's being trialed in people for Nipah, and it's a Hendra glycoprotein, so they're sufficiently conserved. It would be interesting if we needed to get to that point against LAYV. Yeah, interesting point. Troponin is the marker for heart attacks. So it's a damage, myocardial damage, right? Makes sense.
I don't know.
Michael, thank you for your contribution. Really appreciate it. People who are 60 years and over expect some protection, yes, if they receive smallpox vaccine. As we just discussed, you may not have complete protection against infection, lesions. You're going to get infected always, but you may have some lesions, but you may not get more severe disease.
But I don't know that's a different. I don't know that percentage of people is different than people who are naive and then get the vaccine, like my age or younger. So until you come out with the numbers, I don't know that's going to be a big difference.
Are we going to have those numbers?
I don't know. I'm not the people who run trials. I work on enteroviruses. I don't know.
Have you read the paper on Science about the antibody that protects against SARS-CoV-1 2? What potential do you see if so? I don't know which one you're referring to, but Bjorkman's candidate did protect against SARS-CoV-1 as well, right? It induced neutralizing antibodies against one.
Yeah.
And you saw today, you sent me today a SETE paper.
Yeah, it's about T cells.
But that's common cold coronas, right? Okay, it's different.
Protects against, it has some kind of protection for SARS-CoV-2.
Are you okay with the echoing now? Is it done?
I think so.
Okay, so that's a potential issue when you have the stream open on a YouTube channel. It's playing, plus you're hearing me, so you're going to hear it delayed. But now it should be good.
I think so, but I just turned off the whole browser. I don't think it was an open window, but I don't know. It's done. It's gone away, I think.
Besides being virologists, why do you think you've dodged corona so far? Had nothing to do with being virologist, does it?
No.
I have no idea. It's not like I don't get infected. But I really don't know. And the thing is, it's funny that many of the TWIV team have not been infected. I think Kathy and Rich and Brianne have not been infected. And Alan.
Oh, Alan has it?
I think he said he tested positive. I don't remember. Nobody wants to get old. Well, I think some people mind it less than others, right? If you're healthy, it's not as bad, right? Although you just don't know how things are going to go, so you'd rather not. But you have no choice. You just have to. The way I look at it, and you tell me what you think, with age comes accumulated experience and understanding of things, and I think that's useful. I mean, I know a lot of people who are in their 80s have wonderful accumulations of knowledge from their career, and they're wonderful resources. Great people to talk to. And if they're healthy, they can spread that information. What do you think, Amy?
For sure.
Is it worth being worried about long COVID after getting infected while fully vaccinated?
No.
No, it's not worth being worried. And I think,
Didn't your MD lunch party say that she saw very little long COVID or something?
yes, and she said she sees long other things. Well, she sees many different kinds of respiratory infections in her patients, and many of them have long-term symptoms afterwards. But also, Daniel said last week, understanding the frequency of long COVID after infection of a vaccinated person is challenging. We just don't know what the frequency is. It happens, but I wouldn't worry about it. And I don't worry because I don't see the numbers. And someone would say, well, if it happens at all, shouldn't you be worried? No, you can't be worried about everything. You get on a plane, you should worry about it crashing, right? But no, it's a low frequency event. So I suspect that long COVID is a low frequency event, and we just need to get the data to show that.
Yeah, and I'm sure we'll get like a bunch of people who will write in or Twitter and say like we're negating long COVID, blah blah blah.
I'm not
I
negating
love
it
this
at all.
conversation.
I'm not negating it. I'm just saying I'm not
I'm
worried
saying
about
it's
it.
it's overhyped. Move on.
I
Let's
agree.
move
I think
on
it's
now.
somewhat overhyped, but I don't think that's a fair thing to say, because then some people say, well, let's not work on it at all. And that's not fair.
I
We
didn't
need to
say
work on
don't
it.
work on it. I just said it's overhyped. There's a difference.
In this world, nothing can be said to be certain except death and viruses. That's right. Viruses are certain. And Amy is right. We will all in the long term be infected with SARS-CoV-2. Even Amy and myself and all the TWIV hosts who aren't infected. At some point we will get infected, yeah. And probably have mild symptoms, I suspect, because that's what immunity does. Oh, here's a tough one. Would there be any reason someone with eczema should not get an intradermal monkeypox vaccine? I don't know the answer to that.
I have to talk to Daniel.
I do know that it's not recommended for you to get ACAM 2000, which is the other smallpox vaccine, it's a more traditional one, which is given by scarification. And people with eczema shouldn't get that because it can cause pretty severe reactions. But I don't know about this. I would like to ask Daniel. I don't know if, Karen, you can send me an email at daniel@microbe.tv. If you can do that tonight with this question to Daniel, I will put it in the queue for tomorrow because I don't know if I'm going to remember it. I'm going to write it on a sticky right now.
You're going to leave the sticky at home.
Intradermal, so that's Genomes and eczema. No, I'm going to take this sticky, Amy. Look at this. All right, see this sticky?
I see it.
I'm going to stick it on my laptop, which goes with me. That's what that space below the keyboard is useful for on a laptop. You can put stickies on it. What do you think of that?
Okay.
In case Karen doesn't email. All right. Some update on my friend with COVID. His symptoms are gone, but still positive. Despite being with him, I never tested positive, which is strange. I was in a crowded setting. Not strange at all. It's not easy to transmit. Only certain people transmit, and some people don't get infected for whatever reason. A friend of mine had COVID, tested clear, and then got COVID again 4 weeks later. So I don't think you got COVID again. I think that the negative was not a real negative. It was a false negative, and he or she was still infected or had actually.
Well, I don't know if I would say that they were infected. They were just positive in some tests.
Never went away, yeah. Earlier this year, you mentioned there may be some plans to conduct a certificate-paid virology course like on Coursera sometime this year. Are there updates on that? No, I don't have anything to say about that. It's something I'd like to look into, but not right now, no. In New York City, data on monkeypox, there are people over 50 Is anyone finding out if these are people who did or did not have the smallpox vaccine? Yes, I'm certain that we will have those data at some point. Yeah, I don't know of them at the moment. Do you, Amy? And I think if I asked Daniel, he would say, I would like to have that data too, but we don't have them yet. It's probably being compiled, right?
Maybe, maybe not. Who knows in this crazy world we live in?
What happens if after we die we discover that Mel Brooks is actually God?
I guess Anne Bancroft will be really lucky.
Is that his wife?
Yeah, that was his wife. She died.
And Kathleen never saw Star Wars or the producers. There you go. Kathleen is like a mishmash of you and I, Amy.
It's good.
Springtime for Hitler was the play within a play in the producers. There you go, right?
Mm-hmm.
I referred my SARS-CoV-2 positive dialysis patient to our local ER for remdesivir, but the ER doctor refused. He said remdesivir doesn't work on the present variant. Yes, it does. It does. Remdesivir is a polymerase inhibitor, so the variant is not altered in its sensitivity to it. Some of the other monoclonals don't work. But
well, it's not monoclonal, so you can't say some of the other monoclonals. You can say some monoclonals. Why do you have to pick on me?
You don't have to pick on me. You know, I'm making a lot of words here. I'm going to slip up from now and then. Some of the monoclonals do not work. How's that?
That's good.
Molnupiravir still works. Paxlovid still works.
Look, the ER doctor's just ignorant.
Bebtelovimab.
He should review the facts before he gives advice, or he should contact somebody before he gives advice, especially if it's misinformation-based advice. Not good for the ER doctor. Go back.
Go back to medical school? It's not going to help.
Yeah. Okay, well, then go back to some kind of clinical training. I don't know what they do, but go back.
Just listen to Daniel. Daniel tells you what you need to do. But
that would mean I have to know about Daniel, and I'm not sure he knows. Otherwise, he would have given better advice.
So here's the thing. Every doc in the world should listen to Daniel. Okay, he should have millions of listeners. And who has millions of listeners on YouTube? Charlatans who know nothing about viruses or clinical treatment.
No, they don't have millions of listeners. Kim Kardashian's tush has millions of listeners.
I didn't mean her. I meant some other people who can...
Right, but they don't have millions. They just have hundreds of thousands. There's different.
Consider themselves experts, and they're often mentioned by our listeners. I don't mention them anymore.
Well,
anyway.
You can have a big discussion next Friday with your guest about this crap.
I doubt it.
Because he's promoted some of them.
Oh, Andy Slavitt, you mean?
Yeah,
that's the week after. That's next week. That's not this week. Is Mitch Linsky. Yeah, next week.
That's what I said. Next Friday.
Well, you're going to tell me what to ask him.
Yeah, it's not going to be pretty.
Okay.
That's the way it is.
With the benefit of hindsight, what scientific bias do you recognize in yourselves that affected your view or expectations during this pandemic experience?
What scientific bias?
He wants to know how we screwed up. Yeah.
How I screwed up?
You or me. What did we have a bias that clouded our view?
Did I have a bias that clouded my view? A bias like everybody's dumb?
No, come on, be serious.
I'm trying to be serious. I mean...
Did you think vaccines prevented infection, for example? That some vaccines prevented infection?
No, not until you told me originally that the guy who did the super antigen corrected us and said that the HPV did. And then I thought, oh, well, he must know because that's, I guess, true. He knows, I don't really know that much. And then I went to an interview and the lady told me absolutely not. So my initial gut was correct, but I was wishy-washy.
I think that
I think yours is anagenic drift.
yes, you're absolutely right. Thank you for reminding me.
That's what I'm here for.
I thought you liked me.
I'm here for that. Now I'm going to send you my bill.
Yeah,
It's not
I
that
thought
I dislike you. I just think.
I thought coronaviruses did not do antigenic drift. Yeah, that was a bias. It's a good one.
I think the thing is that we always. I think it's not really a bias. I think it's just a misuse of a little bit of a misuse of your training, because you kind of forgot the idea that you just plug the next virus into your system and ask where it differs, and nobody ever did that. Like taking coronas and plugging into the idea of like flu, right? That undergoes tremendous antigenic drift.
Yes. Folks, we have 526 of you tonight. That's awesome. And we have 229 likes. Could you please hit the like button, which helps other people find us, because the more likes we get, the more YouTube will let others know that this is happening here, because we think this information is important. Amy, did you know that searching for pathogens in sewage is a crappy job?
Of course.
I thought you would smile, but you didn't.
I did.
I have a friend who developed Miller-Fisher syndrome after getting a flu shot 2 years ago. Will she ever recover? So Miller-Fisher, if I'm not mistaken, is kind of like a Guillain-Barré.
I don't know.
I doubt it. The only flu vaccine that is associated with Guillain-Barré was the swine flu vaccine of 1976.
Ed Kilborn.
Yeah. So I doubt it. I don't know anything about recovery from it, however. My dog recovered from a canine variant of that. But that's different, I understand.
Luna got a canine variant of the flu vaccine?
No, she got a canine variant of Guillain-Barré. If you're not precise in your language, Amy will get you.
I just didn't understand.
You're just
like,
being honest.
wow.
No, but you know what happened. I told you about it, right?
Yeah, I know.
Did mask wearing increase mucosal respiratory temperature?
No.
I doubt it. I don't know of any studies that looked, but I don't see how it could, because you still have a flow of air in and out, so I don't see how it would increase the temperature, right?
Right.
Is there proof that COVID invades the brain, causing long COVID? No, SARS-CoV-2 does not invade the brain and cause long COVID. There's zero evidence for that. No. Would you say that is there's no evidence for that, Amy?
Yeah, there's really no clinical evidence for it. There's a lot of bad, shoddy in vitro laboratory experiments that are interpreted that way, but what can I say?
If someone wanted to pursue virology in the industry, where could they go? I don't know. What does
that mean?
Industry. Because CDC is not an industry, right? Industry is a company.
No, it's not.
Like Moderna and Pfizer, right? That's the industry. So you go work for a company. You just so I don't know what they want these days. If they want a postdoc or they want you right out of your PhD, Amy, do you know?
Well, right out of your PhD is a postdoc.
Yeah,
You
but
mean
some
after
companies
being
are
a
not.
postdoc?
You know, some companies are not requiring postdocs any longer. They'll train you the way they want to train you.
Well, then it becomes a postdoc.
Anyway, if you want to work in industry, you just apply, right?
Yeah, I
And you
guess
should
so.
find out
I
what they
don't really
want.
know.
You should find out what
I
they
don't
want.
really know. Well, sometimes they hire people who like are not good anyway. So like the association of echoes with IBD, not good.
I had COVID in April, in bed for 3 days, general malaise for 3 months. Symptoms went away. Now they are back. Is this a common experience? So this is a good question. Many people will say they have a similar experience as you do. I don't know the number to tell you because, as I said, it's a challenging number to get at.
Talk to
Amy,
Daniel.
how does SARS-CoV-2 affect kidneys?
I guess it infects them via ACE2, but I don't know that we've ever said that it really affects kidney function.
How about damaging blood vessels, causing clots, and so forth? Is that part of the pathogenesis in the kidney?
I don't think so.
I mean, there certainly seem to be kidney injury in COVID patients, right? But I don't know how the damage occurs, whether it's, I suspect it's not a direct viral damage. I think it may be an inflammatory damage. And I just think people do not know this and they jump to conclusions before they know the answer.
Well, you'd have to do some autopsies and some experiments. Where's Kathy when you need her?
Today, Healthline published what cute cats, look at that. A monkeypox article, and at the bottom they said to get monkeypox virus information from the CDC, the Infectious Disease Society, and you.
That's great.
I mean, who's you? Q&A?
No, I bet you it's TWIP. So let's look it up. Today, Healthline?
I don't know what that is. Do you? Do you look at Healthline?
No, I don't know what it is.
Healthline. So here, 8 questions about monkeypox answered. Let's go to the bottom of the article and look for that. It's a long article. It's pretty, how does it spread? Is it an STI? Is it deadly? Is it curable? Is there a vaccine? Ay, ay, ay. Keep going. No, I don't see a link to us. Nope. Do you, Amy? Not found.
Mm-hmm.
Nope. Maybe they took it out.
Hold a second. You have to keep looking. I have to keep scrolling.
Well, you do that while I move on to the next one. How's that? It's a very long article, but I searched the whole page. I didn't see it. Anyway, that would have been cool, but I don't think it's true. What is it about China that makes it the source of so many viruses? There must be something specific going on there that makes it the first country to pass viruses. No, that's not true. It's not the first country to pass viruses onto humans. But they have passed some on, and some that have gotten a lot of notoriety. And they're looking for viruses, and whenever you look, you find them. But you can discover viruses in other countries as well. Yeah. I don't understand this. Is the secret recipe for a universal vaccine buried in Bedminster? Is that a reference to something?
Jimmy Hoffa is buried all over the place.
As a previous person with long Lyme, which was never widely accepted, there is now broad acceptance of long COVID. Do you think will now be acceptance of long infection issues? I think there are a lot of long infection issues after every infection or many other infections that we don't appreciate. And the reason it's been scrutinized with COVID is because there's so many cases, just millions and millions of cases, right? But maybe we'll be more willing to look at it with other viruses. Yes, for sure.
Stephen Sharf should be removed.
I don't see it.
At the
Why?
bottom, it's offensive. It's all of the gain of function days that are going on at the Wuhan Institute of at the Institute of Virology in Wuhan. It's offensive because it's an indication of supporting the lab leak hypothesis, and it's just wrong and offensive. It should be removed.
Yeah, well, he also thinks our live stream is too long. We'll go. Don't stay then. Most people stay for the 2 hours. We're answering questions for crying out loud, as my father used to say. I never understood that. We're answering your questions. That's why we take 2 hours. So for someone to say you're spending too much time educating people, I think that's very counterproductive. Here we go. Is there a connection between preventive childhood tonsillectomy in those over 50 and impairment of our cellular immunity due to fewer germinal centers? I think not, because it's 2 tonsils at most out of many, many lymph nodes that you have, right?
Yeah, exactly.
So I doubt that they would have any effect. I'd love to see Vincent on the train with a plastic tree. Yeah, that's what I would have to do, bring it in on the train.
It's not happening. Move on from the plastic tray. We're not getting a plastic tray that one dusts. Not happening. End of discussion.
You have to dust live plants too. I never dusted a plant. Have you? Well, you don't have plants. We have a lot of plants inside, but they seem to deal with dust in their own way. I mean, I know that it would interfere with photosynthesis, right? Oh, look at this. Amy was your last apprentice because she's the definitive one. You rock,
That's
Amy.
True. That's true.
You couldn't go. I couldn't continue after you finished, right? I said there's no point. What do you think of that,
That's true.
Dr. Rosenfeld?
It's pretty much true.
Christian Drostin.
Somebody about Gromeyer's repo, where they put the rhinovirus iris into polio.
Yes.
Yeah. I love how something problematic as polio is capable of being useful in this way. I believe that a significant portion of the people died.
Yeah, I don't think that the treatment of glioma with poliovirus has been particularly effective. I mean, I think it extended life in a few patients for a month or two.
Yeah, exactly. I don't think
but
it was.
Can you tell me, Amy, how you would continue to use poliovirus in a post-eradication world?
You can't. No. You can't use capsid proteins.
I don't see the point of even pursuing it. Anyway, do you know that if SARS-CoV-2 replicates at lower temperatures? I don't see any good evidence for that. Do you? Has there ever been anything published?
No.
I haven't seen that.
But he's like obsessed with this concept. He's been obsessed with this concept for like 2 years, and it's just not true.
How has there been so little study on monkeypox, considering it's been known for so long? One word.
Eradication.
No, Africa.
No, eradication of smallpox reduced the amount of interest in poxvirus.
Well, that could
research.
Be too.
But it's the same way as the eradication of polio would have reduced the amount of interest in enterovirus research, except now we have an epidemic of AFM that clearly is not polio related, which makes the idea of eradicating both AFM and polio kind of obsolete.
Is AFM a pandemic, Amy?
Yeah, Tony and David wrote an editorial in MBio in 2019 calling it a global epidemic, which is a pandemic.
They asked me to sing happy birthday to you. Happy birthday to you. Happy birthday to you. Happy birthday, dear Amy. Happy birthday to you.
Thank you very much.
To save my life, but
it's good. It's cute. It's good. It's positive.
Let's see. Why do we look at the flu viruses in the southern hemisphere to determine what is going to happen in the northern hemisphere?
Because it's been shown to be 6 months ahead of us.
Yeah, so they have their winter down there when we're having our summer, and they're having their flu season. And those viruses often show up in the northern hemisphere and to presage what we're going to get. So that's why we look, and they look at us in our winter for what's going to be there for their winter. So there's a lot of global surveillance to do that.
Mm-hmm.
Vincent got me into virology. Thanks for posting Coursera. Gosh, that was ages ago. Ages.
Good.
A friend living in a northeastern Brazil city recently contracted dengue and understands the prevalence has risen. Paul Offit said this is also true in Puerto Rico. Is this a new variant or why the increase?
No immunity. Larger naive population due to
Distancing?
quarantining. And
You don't think it has anything to do with climate change?
well, I'm sure, but it was already in Brazil. And northern Brazil is still at the equator, so it's still in the appropriate climate for the mosquito, and I believe so was Puerto Rico. It's not like between Brazil and then the southern part of Missouri.
If you get exposed to a vaccine-derived strain of polio, your gut will become immune afterwards, will it not, whether you have IPV or not?
Yes.
Yes, but just remember that
It doesn't replicate as efficiently.
The immunity will decrease and you'll be infected. You'll have a memory response, which takes a few days, in which time the virus will have reproduced and been shed. Yeah. The UK is offering both booster and catch-up for those that missed. London has a higher level of vaccine holdouts similar to New York, some areas. Yeah, I understand that. Yeah, and in Rockland County, they did a similar thing, right, Amy? They did. Here we're putting up vaccine places, yeah?
Yeah, they're still doing vaccine clinics in Rockland and Orange County, and now the CDC is out there.
How dangerous is the virus in China? We talked about it earlier in case you missed it, but at the moment there's not a lot of concern. But we can always be wrong, right? We were wrong about monkeypox. We don't really hear about the spread of SARS-CoV-2 in underdeveloped countries anymore. Is there a rise in numbers or deaths in non-Western countries? Well, the numbers...
You have to go to the Hopkins site. I don't know.
I don't think there's a rise, but you can get numbers from these countries. However, we always wonder if the reporting is that good. Many countries don't report. So, you have a lot of countries with very few cases, but that's just because they're not reporting their cases or they're not testing at all. So,
well, one could argue we have numbers. How good is that reporting? We have numbers. What do our numbers actually mean?
I thought it was 10-year gap for yellow fever booster. That's correct. If you get yellow fever vaccine, you should get a booster 10 years later. That's correct. But I'm just not certain that an infection is, as contrary to what Amy says, I'm not sure the infection gives you, in every case, good immunity. So you might as well...
Well, vaccine doesn't give you every case good immunity. So yeah. I don't know what the argument is.
So combine them and you're good to go, no?
I would think so, but I'm just saying, that in general, I don't think you should ever assume that immunity from an infection is less effective than immunity from vaccination. And again, the guy who was with the super antigen, told me I was wrong, and then I turned out to be right. So,
I'm getting a fourth dose, fifth if you count an ancestral infection, and handstand. I'm no harm done with multiple vaccines. Not that we're aware of. No.
No, it's fine.
John Udell indicates in TWIV 597 protective action of fever. I think this is a theoretical idea, right? That fever protects against some viruses, but we don't know what the mechanism would be, right?
Yeah, I don't know that I would say that was correct.
I learned about BA 4.6 today. It's circulating. It's another Omicron subvariant. BA 4.5 is still dominant. This seems to be accelerating. We'll see what happens. I think this is going to be the story for some time now. A&V, are the HPV vaccines not an example of a vaccine that gives better immune response than infection? Durable B-cell immunity and antibody titers between 1 and 2 logs higher than infection.
Well, what does antibody titers mean?
Well, I think that's a good question.
And how do I know it's mediated solely by a durable B-cell response and not a T-cell response?
Mm-hmm.
I don't know. I don't think that this is necessarily right.
But that the vaccine gives better immune response than it gives a better antibody response, but we don't know what. I mean, they also be giving a good T-cell response, right?
Right. I don't know what that means. You could have you could have a high titer antibody response, but I don't know what that means.
Why are inactivated virus vaccines less effective than single protein-based vaccines? I would have thought being exposed to the entire range of viral proteins would produce better immunity.
And activation always has the potential of altering the antigenic sites so that the antibodies that recognize them actually don't do anything.
Yeah, that's basically it. That these are old methods of inactivation that really trash the antigens. And we're fortunate with an inactivated polio vaccine that it's still really good after formaldehyde inactivation. Isn't that?
There are some studies by Miner that question how good it is.
It's being used in the U.S., Amy. It works. Prevents polio, right?
Just because it's used in the U.S., I wouldn't have used as my answer.
Have you seen
it's not like we're a bunch of geniuses.
You've seen any data on Peter Hotez's vaccine and how effective it turned out to be? No, I haven't seen any data on it. Have you?
No.
We know nothing about it. I got yellow fever in Riverside, California. The mosquitoes that transmit yellow fever have been found as far north as Sacramento. Wow. At one point, they were up to Philadelphia in the 1700s. But not on the East Coast. They're really just in Florida.
Well, Philadelphia was on the East Coast. It still is.
Did I say it wasn't?
Oh, I'm sorry.
My son got a booster, but you can be funny. It's fine. My son got a booster for meningococcus. He developed side effects, headaches, weakness, neck pain. Is this a common reaction? It's a reaction. Would have to look.
I have no idea. It's a bacterial disease.
But it's in a vaccine, right? And you're going to get inflammation like you do with many vaccines. And it means that you're responding. And you can have headaches. You can have muscle weakness, neck pain. Could be. Is there any other virus that would affect taste and smell in the way SARS-CoV-2 did? Yes, you can have taste and smell losses with other respiratory viral infections like influenza virus. And again, because of the high numbers of SARS-CoV-2 infections, it was amplified and got a lot of attention. But there are plenty of people who lose it with other infections and you never hear about it. Could the mRNA vaccines be also given a fifth dose intradermally. Well, you'd have to test it, right? You'd have to see if you get the similar, and there's no shortage of the mRNA vaccines, so there's no need to try and conserve it in that way, unless intradermal gave you a better response, but the intramuscular is really good, so I think not.
I don't know that I would assume that intradermal would mean that you could always use 1/5 of the dose.
It's just limited by the volume you can put intradermally, you know?
I guess.
Yeah.
But you might have to, so you might be limited by the volume, but you might have to have like far more administrations. Right.
Yeah.
I don't know.
Brother got 4th vaccine in June because told could still get the 4 or 5 booster when it's out. Now saying will be out in September. Earliest it would be safe for him. 90 days.
To do what?
To get the 4 or 5 booster in the fall. Is imprinting a concern? Well, that's what Paul Offit suggested. You're going to
I don't
mainly
see where there's any data
there's
for
no data on it, no.
original antigenic sin, which we've now recoined as imprinting.
I kind of like imprinting is a better term, don't you?
Sure.
I don't think we should use religious references in disease terms. Do you?
I don't think we should do a lot of things that we've done in the past, but you know, they don't ask me.
Was just listening to a podcast on tetanus. That's one example where the vaccine has a much greater immune response than infection. Bacterial infection doesn't produce immunity to reinfection. Okay. I don't know much about the tetanus. Tetanus is a toxoid, right? Vaccine. It's a purified
it's a toxin.
toxoid, which perhaps in a bacterial infection, you don't make much of an immune response to it.
Could be. I don't know.
I wonder if Paxlovid rebound, I know, terrible name, is any connection to those who get a reduced dose due to kidney function issues. I have not heard of any such connection. You could suspect, but I mean, the data might be there, right, to look at it, but I don't know that anyone is doing it. Does fever reactivate herpes viruses? It can, yes. Many people, upon getting a fever from a consequence of some other infection, have a reactivation of herpes and get cold sores. Yes, absolutely.
Yeah, but that's just correlative, because there's a lot when you get something else, you always have an immune response and some other stressor. So fever might just be hanging out and have nothing to do with it.
It's one of multiple things that could be causing the reactivation, right?
Right. So, you know, I wouldn't have said it causes. I would say could fever correlate with a reactivation? Okay. Yeah, associated with, because causes sounds like it's the sole thing and it's a proven thing, but it's not. There's a lot of stuff when you get another infection. Like we see like shingles reactivation after a COVID vaccine.
Yeah.
Right? Wasn't that the thing? Well, was it due to the mild fever that got you that reactivated the shingles, or was it due to the fact that, you know, vaccination was a stressful time and blah, blah, blah?
I agree. There are a lot of things happening when you have a fever. Might be other things as well, or a combination. Yep.
Yeah, I think it's more honest to say it associates.
What's the difference between SARS 2003 and COVID? Well,
tons of stuff.
SARS 2003 stopped after 8,000 cases. It didn't do community transmission. Most of the peak of virus shedding was at the peak of disease, so there was no asymptomatic shedding. And mostly you were in the hospital at the peak of viral disease, and that was easier to control the transmission there.
Yep.
You're not usually allowed on the mat if you have open sores, right? But you can imagine that you have a sore that's covered by your singlet, and then maybe it gets pushed aside and in the activity, and then it rubs the other person, right? Yeah, so can happen. I think we have to worry about that. I just went through COVID thanks to two Pfizer and Moderna boosts. I had a bad cold with full recovery. A coworker lost sense of taste and smell. I'll count myself lucky. Yes, I know people who have been multiply vaccinated get infected and lose their sense of taste. Yes. Yep. Oh, here's an interesting one. It's official. BA5 is weak sauce, and Bob Wachter and Eric Topo Chico Topol are full of it. Well, we've said here for a while that they are.
For sure. I don't see what am I going to do with this information? It's not going to change the fact that, they have damaged certain things and they continue to do it. So
does your gut tell you these COVID variants are getting pathogenically weaker over time, yet more transmissible? No, my gut doesn't tell me that at all. I think the population immunity is rising, and that modifies disease. And people are going back to their lives, and it's human behavior that is spreading infection. It's all about humans, Amy.
I don't know anything about that, Vincent, since I think we wrote an editorial that talks about, it's this human behavior is more concerning than the variant.
Than any variant. Yep.
Why write an editorial and everything and be an expert and spend your whole life studying this stuff if, you don't know anything? It's a good plan.
Don't give up, Amy.
I'm not giving up. Just saying I'm just being sarcastic about it, being a good plan.
Is this long-term, lifelong immunity, which we talked about for smallpox, conferred by memory, B-cell activation, B-cell and T-cell, yes. Memory cells, for sure. Yeah.
But see, this is the problem. Like, everybody just focuses on memory B cells. It's like I talked to the flu guy in the office over like the last 3 weeks, and you know what I told him?
I don't.
Okay, so any answer to any antibody question, any immune question, any structural question, anything of importance to polio, guess what it is? The answer is
antibody.
No, it's always the BC loop of VP1. It's the BC loop of VP1. It's the VC loop of VP1 for any enterovirus, and that's like not even true. But no matter what, it's always the BC loop of VP1.
So what Amy is describing is an antigenic site on the viral capsid, right? Do you want me to show?
It's not just an antigenic site. It controls species tropism, right?
Do you want me to show a model of it?
Sure, but you do have to admit it controls species tropism.
It does.
It's not an antigenic site.
It can. I have no idea if I had a polio vaccine. I guess if I was born in the early 60s, I would have been. Yes.
If you went to public school, you had a polio vaccine. You're not allowed to go to public school without it, basically. There are exemptions. But
Ray,
not really.
Thank you for your contribution to science communication. Have you heard of someone using bebtelovimab and Paxlovid? I have not. I don't know why you do both, right, Amy?
Yeah, I don't know, but Daniel probably has an idea, considering the fact you can only give one dose of each, right? So one course.
Yes.
I should say not dose, one course of each. So I guess if you got Paxlovid and then you got rebound, and it was really not, I don't like the word rebound, but you know what I'm saying, then I guess they would come in with the antibody. I don't really know. Talk to Daniel.
So I haven't heard of it, but Daniel will be the one.
Talk to Daniel.
Don't get stern, young lady, okay? How hard would it be to create a virus from scratch using genetic sequence? Some are worried that smallpox could be created in the lab using the sequence. Yes, that's why we have a strategic national stockpile comprising 2 vaccines and 2 antivirals for smallpox.
Wow.
Because you can't... just a minute, let me finish. There was a paper published a couple of years ago where they took the sequence of horsepox virus. Virus is extinct. We have the sequence. They had oligonucleotides synthesized. They had the genome synthesized. They put the DNA into cells, and out came the virus. So it can be done. And so that's why
Yeah, so can it be done for polio too?
absolutely.
Eckhard Wimmer.
It's
But
not easy. You need a lab to do it.
I was just about to say, like, do you know how many failures there were for that one experiment? I'm still, it's like there's huge amounts of websites that tell you how to build a bomb in your mother's kitchen. How many successful bombs have been built in your mother's kitchen?
I don't know about bombs. I don't know if bombs are a good.
It's true though. There are hundreds of them. That's why we have you take off your shoes and all the TSA crap, and yet not a single one of them has really been successful and as that have been built at home. So yeah, you could possibly do it, but you know how difficult it actually is. It's like unbelievably difficult. If it were so easy, my people could make infectious clones tomorrow.
Do you think Covaxin has any benefits? It is a whole virus inactivated vaccine. So I have limited familiarity with the data. So the data from China on their 2 inactivated vaccines, they're not terribly good unless you follow with a mRNA booster, which is a paper we did on TWIV, which had been sent by Amy.
Yeah, but that just demonstrates that the formaldehyde is toxic to the antigenic sites.
Yes.
So
I think that's what Amy is saying, that I doubt it's going to have a lot of benefit. However, there are a lot of people in the world who need to be vaccinated. That's one of the reasons they cranked out this vaccine, right? Old technology, they could do it very quickly.
Really? Because I could have sworn we both know a patron virologist who swears every time you talk to her, that's what she says. In Wisconsin?
Yeah, I know. I think the inactivated polio vaccine kind of spoils the show, right? Because it really works well. I know you think it doesn't work so well, but it really works well.
Well, it works really well, okay? However, there were at least 11 attempts to do it before Salk was able to do it and get it successful. And then subsequently, there have been multiple attempts through, 30 other viruses where it was completely unsuccessful. So when you take the numerator of 1 as success and you go through all of the failures, which is probably like at least 60, 1 out of 60, not a high success rate. Do the math. Not good.
Anyway, Covaxin, I'm suspicious that it's going to be very useful. I mean, if you give limited immunity in a population and you have to come in with a booster, it's not very successful, is it? Nice to see you quoted in the Wall Street Journal. Amy, can you send me the article? Because I can't read it. It's behind a subscription limitation. I didn't see it. All right, Chris wants us to debunk the term Paxlovid rebound.
It's not rebound. It was observed it's been observed for people who were not treated. So it's not a rebound.
It's just a continuation of the infection. In some people, they don't clear it within the 5-day period, and that's the way the virus is reproducing. It has nothing to do with Paxlovid at all. It's a feature of a natural feature of the infection, I would say.
Exactly.
Maybe you were infected but were asymptomatic and so didn't test. Well, it's possible. Both Amy and I went through a lot of random tests at Columbia, PCR tests, and we're all negative. And then most recently, to get back from Zurich in May, I had a test negative. But yes, they were all either PCR or antigen tests, and I've never done an antibody test, so it's possible that it might be positive, but I don't think I'm going to do it. Because what would I get out of it, right? Just to say that I have been infected? Not sure that's useful. How can you not be worried? Isn't the frequency 1 in 3, 1 in 4, 1 in 5 of long COVID? No way. It's not any of those. I'm sure it's 5%.
Well, that wouldn't make any sense. That wouldn't make any sense.
Absolutely not.
And it's subjective anyway. Your questionnaire will be different from my doctor's questionnaire. So
I don't have skepticism of long COVID, Dave. I am suspicious of the high numbers. 50% of infections after vaccination give rise to long COVID? No. This is the way that studies are being done. They're not appropriate. Using hospitalization, for example, as a metric is not correct. Sorry, that's the wrong term. Using fatigue as a main metric. There's no clinical diagnosis of long COVID. There's no way to precisely say someone has it or not.
that's what I just said.
It's often done by self-reporting, which is flawed. It's often done by an app, which eliminates people that don't have apps. So it is, as Amy said, it's a flawed way of measuring it. That's what I'm suspicious of. I don't doubt that it happens. So please don't put words in my mouth. Don't say I'm skeptical of it, because I agree that it occurs. I just don't know the frequency. Thank you, Magpie, for your contribution. Our whole house had COVID over the 4th, all fully recovered. Should we get SARS-CoV-2 boosters in the fall with our flu shots?
No.
No, if you've already been vaccinated, you don't need boosters after having been infected. No, not necessary. Tested positive 2 days ago. 2 times vax, no body aches, fever, just mild congestion. Is lack of more serious symptoms any indication that the immune system is or isn't fighting? No, not at all. You have mild symptoms. Your immune system is working. It caught it early, and it's retreating. Always have strength and joy. Wow. Had COVID in June, also March, vaxxed and boosted. I had asthma both times. So respiratory symptoms were rough, and the cold flu type symptoms were the worst the second time around. I assume you stayed out of the hospital. You didn't have severe COVID. That's the vaccine working. Thank you, Kip and Laura, for your contribution. I look forward to the dining pleasure and accurate knowledge feed you two experts provide each Wednesday evening. Thank you. You're welcome. Amy and I are pleased, and I'm still stunned that someone complained that we'd spend 2 hours doing this when we're trying to help out. Isn't that weird, Amy?
Very strange.
So a University of Michigan hospital doctor said you carry the infection 90 days so you are not being reinfected. This is totally wrong. You do not carry the infection for 90 days. Listen to Daniel Griffin. 1 to 2 week course. Everything beyond that is not an infection. There are certainly chronic health conditions that would have occurred naturally in the absence of a pandemic that will have been conflated for long COVID. Difficult to parse out from legit long COVID. Yes, that's part of it, I think. Yes. But I also think that the numbers have been, with the exception of MECFS, right, which is affecting a lot of people. I think it's always hard to find these long-term after effects because numbers are low and no one does a study to parse it out. But you have a physician who says, yeah, my patients have a respiratory syncytial virus infection and they have symptoms that linger for months. That would be long RSV, right? But we don't study it. Here's one for Amy. Do you think the general population should get vaccinated against monkeypox?
The general population? Sure.
Really? You think so?
Sure. Why not?
Like you and I've been vaccinated for smallpox. I don't need to be vaccinated. I have low
right,
risk.
but I have not.
But the thing is that, it is not a gay disease. It is not an African disease. It's an infectious disease. So at risk is very difficult to parse.
Well, he didn't say at risk. He just said at general population. So why?
I think anyone who wants a monkeypox vaccine should get it. The people who are at risk know who they are. If you, Amy, said you want a monkeypox vaccine, you should get it. You having trouble hearing?
No, my AirPods died, and then I don't know. The other ones don't really work so well right now. I don't know exactly.
I have a couple of Amy comments. Amy demands facts. That's true. She's well trained. As far
Well,
as
what do
I'm
you think I'm
concerned,
going to demand?
Amy is straight up cool. She does not suffer fools lightly. Yep. What's the risk
Well,
of taking
who wrote
Paxlovid?
a thesis and said, this is not creative literature class. What do they expect? Some days it's
Um,
like, really?
what's the risk for taking for more than 5 days? There probably isn't a risk, but the EUA, the FDA said you can use this under emergency use for a 5-day course. And so I understand that people break the EUA all the time, but that's what the FDA says. Amy works for the FDA, so we cannot not listen to them.
Don't bring me into it. I don't have anything to do with it.
You're on the live stream, Amy.
I know, but I'm not. I don't do those kind of things. I run a lab.
Do listen to Daniel. It pays to be informed. There are many hospital doctors who do not know how to test for monkeypox, even in New Jersey. You know, they
Well,
have. Go ahead.
not shocking.
Why is the EUA for Evusheld so strict? I don't know. It's a good question. Maybe they're worried about supply. I don't know. I don't know the reason. Do you know, Amy?
No idea.
Andy Slavitz gave him a steroid plus zinc plus an antibiotic with his COVID treatment. Please ask him what his doc gave him. Oh my gosh.
Sounds like a good doc. Sounds like a genius. So that's like somebody who wanted to give a steroid to my cousin when she was positive and had pseudo uncomfortable symptoms. And I was like, tell him to stay in his lane because you don't want to do that. Luckily, she listened to me.
Is it normal to catch a flu-like infection every season? Is it normal? For some people, it is. Some people get a flu-like illness every season, and others do not. So, because humans are all different, and so they react to infections differently. And I rarely get any flu-like illness. You changed your speaker because now I'm echoing, Amy.
Because my AirPods aren't working. I don't think.
Oh, so you're listening through your computer. Can you lower the volume so that it doesn't echo, please? Thank you.
Lower everything. I don't understand. Technology is overrated.
There are 2 dentists listening. Richard, I often hear from Richard, DDS. Good to hear from you. Good to see you here, and I'm sure you're concerned about monkeypox as well. We'll have to get you new earbuds, Amy. Okay?
Sure.
Would you like a wired pair like I have here?
Yeah, that's good.
I'll send you some this week because we need to have you properly teched out, right?
Yeah, me, high tech.
Okay. Just a point of fact, PCR was invented and developed in industry, not academia. Oh, right. You want to argue about what was developed where? Do you want to argue about the foundations for PCR? Do you want to argue about the enzyme that made it possible, that was discovered in an academic lab? Please, Stephen. Academic research is the foundation for everything.
Well, he doesn't really know very much about the history of PCR. It's not even worth discussing because he's just ignorant.
Okay.
Coming from somebody from Wetmer's lab, what do you think about that?
I think you're right. I have no doubt. Yeah. I mean, I do disagree with you on several things, but not that one. All right. CS says, wait. You both say no vaccine prevents infection. Does that mean they only protect against development of severe disease?
Yes.
So we've been saying for 2 years now, yeah. Here's the article. Monkeypox myths debunked by health experts. Control F this week in virology. Oh, maybe I didn't do the right thing. I Control F TWIV. So I'll have to try it again.
All right, I'll test it out. All right, let's see.
T. Klepper, thank you for your support. Really appreciate it. Really appreciate your support of science communication. And we have 5 minutes left, so we're going to roll down here. There's a lot of health inequality in the world, and there were hundreds of cases of monkeypox infection in Africa for years after the cessation of smallpox vaccination, and very little was done about it. I like this. Take as much time as you like, for crying out loud. I'm nursing COVID arm after receiving my second booster. Thanks for clarifying monkeypox protection from my 1960s smallpox vaccination. All right. Sean, thank you so much. This is for Amy's birthday fund. Is that good?
Yeah, it's very nice. I'm happy.
Hey, we have 390. Sorry, we have 390 likes. Let's get over 400, please, folks, before we wrap this up. We got 570 people here, which is really cool. Like button, please. This is an interesting question that why wasn't Paxlovid tested on vaccinated people? Because you want to know how it works against someone who is not vaccinated. That was the initial requirement, because we weren't certain that the vaccines were going to be available so quickly. And Amy, is that totally fine to have it tested only in unvaccinated people for an antiviral?
For sure. Why you think the virus is? What's the difference?
No,
why would you test it on? Why would you expect an antiviral to work differently in a vaccinated person than an unvaccinated person? You think the virus knows that you're vaccinated or unvaccinated?
Yep. How do I find
seriously, you think that the polymerase works differently? Says, oh wait, I'm going to be in this other environment.
No. Charlotte wants to know how you could tell if you were vaccinated. Well, you
could get an antibody test.
You could have an antibody test
to see if you had a titer.
But I don't know that you could get that nowadays. If you don't know, then you might not have been, and so if you're worried, you should just get vaccinated again. I mean, you could see if you have a scar on your arm, right?
Not everybody's scarred.
But not everyone's scars, I know, yeah.
So like my mom and her two sisters, they've all been vaccinated against smallpox. Only one of them has a scar. I don't think you have a scar on your arm, do you?
I don't know. I have to look. I'll look later, okay? I'll let you know.
Okay, report back next week.
Pardon my ignorance, but is polio vaccine administered as part of a standard package that child needed for school? Yeah.
Yes.
It's with DTaP, right? Diphtheria, acellular, pertussis, polio, right?
Yeah.
Okay. Yeah, some people don't have scars. That's absolutely right. Let's see who else I have to thank for our contributions here tonight. All vaccines. Yes, I got a 10-year-old Tdap vaccine. Yeah, there it is, Tdap. Patricia, all vaccines should be no cost regardless of insurance status. I agree. I think you shouldn't have to pay for vaccines. Does the fact that we don't have much pox vaccine mean we are not prepared for a bioterrorism event? So it's a good question. I'm going to ask Michael Merchlinsky Friday, because I thought we had enough to vaccinate everybody in the U.S., but apparently not, as you can see from the monkeypox fiasco, right? So I want to find out why we don't, because I was under that impression that the Strategic National Stockpile would cover everyone. Can monkeypox be transmitted in swimming pools? Daniel said no. The
Why would the chlorine not kill the envelope?
Yeah.
That's what chlorine does.
Yeah, chlorine would take care of it. That's right. I'm unvaccinated. I've been around positive COVIDs. I've gone to crowded events. I haven't gotten COVID. Should I get this shot?
I would, but
Yeah. Making explosives is not the hard part. Not doing it without blowing your head off is. I would say the same for making a virus, Amy, right? It's both hard and you have to prevent you from infecting yourself, right?
Yeah, for sure.
Robin, and thank you so much for your contribution to science education. What's necessary to make take-in food safe from monkeypox?
You don't get it from food.
You're not going to get it from food. You're not going to get it from casual contact with the container. It really requires some substantial skin-to-skin contact. Could we show you the study on Paxlovid? Well, it's out there published, and Daniel has mentioned it on one of his pods, so I think you ought to go there and get it. Thank you, Ray, for your contribution. Really appreciate it. Incredible results with bebtelovimab. Great. Second day, that's important. You need to get it early. Absolutely. All right, let's wrap this up. Let's get to the end here. Sandra, thank you for your contribution to science communication. John, thank you so much. Wishing Amy a piece of cake, glass of wine, or any treat for her birthday. That's very nice, Amy. Isn't that kind?
Thank you very much. Yeah, it's very nice.
Thank you, Dominique, for your contribution to the incubator and science communication. I have a fruit fly flying around here. Thank you, R. Minon, for your contribution. Really appreciate it. And thank you, James. Happy birthday, Amy. Thank you, James, again for your contribution. And thank you, Chi, for your contribution to science communication. It's great to see everybody here tonight. We had over 570 people. I want to thank the moderators. Once again, we had Les, we had Steph, we had Tom, we had Frank, and we had Vanity Nutrition. Thank you all for your work here tonight. It is rather thankless, I know, but we appreciate it. I want to thank everyone for coming. If you didn't get your question answered, come back next week. We're here although next Wednesday is a challenge because I am in Ithaca, so maybe we're going to have to do it on Thursday, Amy. What do you think?
Oh, okay.
I'm in Ithaca on Wednesday.
Yeah, no.
And sure. I'll be back for Thursday evening, so we'll talk about it offline. But come back next week with your questions. Amy, thank you so much for joining me tonight. As always, I do appreciate it. And happy birthday once again.
Thank you very much.
And everyone stay safe. Until next time.
Good night.
Good night, folks. Bye-bye.